Once the genetic code had been worked out, the next obvious thing you wanted to know was, what is the actual sequence of bases along a DNA molecule? The genius behind developing this method was the biochemist Fred Sanger at Cambridge University. Fred was in Cambridge when I and Francis Crick found the double helix. At roughly the same time, he published the first sequence of a polypeptide chain, the sequence of amino acids in the protein insulin, hormone insulin, and for which Fred, a few years later, received the Nobel Prize in chemistry. When Francis Crick, after the double helix, the British government built a new laboratory, laboratory for molecular biology, to which Crick and Perutz and Kendrew moved, and they asked Fred Sanger to join them. And this was a very wise decision. Fred thought like a chemist and was a very determined man. If you talk to him, you think he's so modest, he couldn't really be that bright. But he's so very bright. And he worked out a way which was really relied on biochemistry to sequence the molecule, use enzymes, and a clever trick. My colleague Walter Gilbert at Harvard essentially devised a method of pure chemistry for sequencing DNA. Now, the first molecules that were sequenced were those of viruses, because you isolated the virus, and from that you isolated their nucleic acid, and you had a pure DNA molecule. Normally, if you looked at, a human cell, so much DNA would be broken, you couldn't isolate a nice single molecule, at least initially. So Fred thought in terms of viruses, and by 1975 had done about 5,000 bases in the small bacterial virus 5X174. And after that initial success, they moved to longer and longer DNA molecules from larger viruses. And here at Cold Spring Harbor, we sequenced an animal virus called adenoviruses, which had about 40,000 bases, and that required 2 people working, and we did it in conjunction with scientists in Sweden. And it was about 1,000 bases a year. Now, once you had it, you could actually look and see which proteins and the nature of them that were coded by the DNA.
Il genoma umano è la serie completa dei geni dell'essere umano, l'insieme di tutte le informazioni genetiche depositate nella sequenza del suo DNA. Essendo contenuto nei 23 cromosomi che si trovano nel nucleo delle cellule, possiamo immaginare il genoma come un grande libro diviso in 23 capitoli. Ogni capitolo contiene alcune migliaia di paragrafi, che sono i geni, ognuno dei quali è responsabile di una caratteristica dell'individuo. I paragrafi sono composti di parole chiamate codoni, a loro volta scritte in lettere chiamate basi. Le basi sono quindi l'alfabeto genetico. E sequenziare il genoma significa comprendere come queste lettere vanno a comporre il lessico degli esseri viventi, in particolare dell'uomo. A differenza del nostro alfabeto, che comprende più di 20 segni, quello del genoma si limita a 4 basi: adenina, citosina, guanina e timina, che si combinano su lunghe catene di zucchero e fosfato, la doppia elica del DNA, e non conoscono segni di punteggiatura. All'interno di un gene non c'è soluzione di continuità tra le basi. Gli stessi geni non sono ordinati per argomenti o funzioni, come i paragrafi di un testo, ma il loro ordine all'interno dei cromosomi ci appare casuale. Sequenziare il DNA, il grandioso obiettivo del progetto Genoma Umano, significa allora scoprire il codice che garantisce il funzionamento e la riproduzione della vita umana.
About 10 years were occupied by sequencing viruses. And the talk began to go, there began to be talk, could we ever sequence all the DNA in a human cell? And that's 3 billion. So 1,000,000 times more complicated than Sanger's first molecule. I was sort of not interested. It just seemed too far off to be necessary, though the sort of DNA technology people began to hold meetings beginning in 1983 to discuss the feasibility of sequencing DNA. And we were aware of it. But the matter was really brought to our attention when Renato Dobecco, the Italian-born molecular biologist, came to give the dedication speech when we had a new cancer laboratory. And he urged that we do the human DNA sequence. He said that if we want to understand cancer, we have to know all the DNA, all the human genes. Otherwise, we'll be dealing with an incomplete and maybe dangerously incomplete knowledge of human cells. I was the one who invited Renato. We had been friends for a long while. We first met at Indiana University when I was a graduate student. Salvador Luria had brought him from Italy. They were both students of Professor Levi in Turin before the war. Dobecco had served with the Italian army in Russia. Luckily, he came out of it alive. And whereas Luria, being a Jew, had left Italy, then fled France and came to the United States when the war had just started. Dobecco and I always remained close friends, and Dobecco was the first person in the United States to sort of take an interest in the biology and then molecular biology of cancer viruses or tumor viruses which, when they infected a cell, they didn't produce more virus, but made the cells cancerous. So it seemed a simple system, and Dobecco focused on a very small cancer-causing virus called polyoma, which if you just said how big is the new DNA molecule, you could say, well, maybe it only specifies the information for 3 genes, at most 5. So it seemed the simplest system to study cancer. And when I started cancer research at Cold Spring Harbor, the first 3 people I hired were all postdocs working in Dobecco's lab. So when Renato said sequence the human genome, one should take it seriously. And 6 months later, we had a big sort of human genetics gathering at Cold Spring Harbor, and we had a session, should we sequence the human genome? And so most people said, no, it's too soon. You don't have the technology. But Walter Gilbert, who had worked out a DNA sequencing technique, which led to both he and Sanger getting Nobel Prizes for it. He said we should do it, and we could do it for $1 a base pair. And since there were 3 billion base pairs, that was going to be $3 billion. Now that's a sort of sum that would almost build a big nuclear accelerator, a physics machine. But we said the amount of money, it would take us 15 years to do it. So it'd be 2% of the money spent by the National Institutes of Health on medical research. And that would $200 million really would be, you wouldn't notice it. You could do the Human Genome Project and the rest of American science could go on as if you weren't doing it. So I was worried, not that we wouldn't do it, but the wrong people would lead it. And then there were two questions, you know, where would the money come from? And by then I had developed, you know, some friendship with members of the United States Congress. And after we had a National Academy Committee, National Academy of Sciences Committee say, yes, you can do the Human Genome Project. It's a feasible project. And we said $3 billion, 15 years. I went to Congress and met a few people. And they voted $30 million for the first year. And once we had the first money, then the question came, well, who would lead the project? And Jim Weingarten, the head of NIH, asked me to do it. When I had been, you know, working to get it started, I never thought I would lead it. And I said, well, I can do it if you let me keep my job in Cold Spring Harbor, because I have a son who's sick and we can't bring him to Washington. So I'll need to be in both places. And since Congress trusted me, I became the head. And my job, I saw, was just get the project run by intelligent, bright people. And it didn't matter that we had enemies who said we shouldn't do it, as long as the people who were doing it were among the best scientists or among the best molecular biologists in our country. And I first visualized it as a project dominated by people. You might almost have to have a small genome city. But it turned out that it later became dominated by machines. That is, we replaced the people who would do 6,000 bases a year by machines which could work much, much faster. Within another year, the machines took over and everyone moved to machines. Gilbert didn't join us. He thought the Human Genome Project would be most efficiently done by a company. He had started a biotech company to use the techniques of recombinant DNA. That was successful. For a while he'd been its president, so he thought, why not a new company? But he couldn't find people to put money into it because they didn't, you know, it was just too far off. 15 years, and they didn't quite see how it was going to make money. So it became a public project, and the question came up soon, once you get DNA sequenced, should you be allowed to patent it? And I didn't like the idea. My boss, then the head of NIH, Weingarten, was gone. She said I was virtually unpatriotic for America not wanting to commercialize on the Human Genome Project, and she fired me. And that was a good thing for me. After I ceased to be the director of the Human Genome Project, it was led by the medical geneticist Francis Collins, who had become famous for being a sort of co-discoverer of the gene for cystic fibrosis. He was at the University of Michigan. We were supporting him using genome money, and it was a very wise choice, Francis. So when we started, we were giving money out to about 10 different labs to let them compete with each other and see who could do it the best. And we decided in the beginning that we would focus first on making a very good physical map of the DNA. We would, along a given chromosome, we would isolate units of DNA which contain several 100,000 bases, and then we would sequence these small things, and we would have them overlapping, and then we would finally put the whole thing together by sequencing relatively small pieces. We didn't know how we could locate the DNA on chromosomes, so we had a library of DNA fragments going from one end to the other end of each of the 24 human chromosomes, the 22 pairs of autosomes and the X and Y chromosome. And so we were basically trying to put together the DNA chromosome by chromosome. A very key step forward was the British government didn't then have the money to join the Human Genome Project, but in 1992, the Wellcome Trust, a philanthropic foundation in England, which was very rich, they decided that they would support a very big laboratory in Cambridge, England, or outside Cambridge, which became known as the Sanger Laboratory. And so they put enough money in, so the Americans were doing roughly 2/3 of the genome, and the British were doing 1/3. And they built this genome lab unbelievably fast. Within about 1 year, they were working day and night, and once completed, it was the biggest genome center in the world, and remains the biggest and certainly one of the best. The aim was to keep reducing the costs, and that meant better machines. And there was a big jump in the efficiency of the machines, which were made by a American company called Applied Biosystems. They came out with a sort of radical new technology in 1998, which would, say, let you move 10 times faster. And so one began to smell the fact that maybe we could do it. So it was all preparation with very few final results, but there was one day suddenly a complication. Craig Venter, who was initially had been at the National Institutes of Health and a very early genome sequencer, formed a private company to sequence the genome. And he said that he used new technology and essentially told the international effort, don't waste your time competing with me, sequence the mouse.
John Craig Venter nato nel 1946, è considerato uno dei maggiori scienziati del XXI secolo. La sua carriera comincia come docente universitario presso l'Università di Buffalo. Nel 1984, però, Venter lascia l'accademia per lavorare come ricercatore al National Institute of Health. Qui comincia a dedicarsi alla sequenziazione del DNA. La decodifica dei geni degli esseri viventi compie progressi straordinari grazie al lavoro della sua equipe di ricerca, tanto da indurre lo scienziato a trasformare il genoma in un business. Nel 1998 Craig Venter fonda una società, la Celera Genomics, per sequenziare il genoma umano con tecniche e strumenti innovativi e creare una banca dati genomica utilizzabile solo a pagamento. Lancia allora la sua famosa sfida al progetto Genoma Umano, proclamando che entro il 2001 avrebbe terminato la mappatura. Il governo statunitense reagisce accelerando i tempi del progetto pubblico e alla fine i due concorrenti arrivano insieme al traguardo. Da allora il celebre biologo non ha mai smesso di sorprendere il mondo con scoperte sensazionali. L'ultima, del 2010, annuncia l'era della vita artificiale. Venter è riuscito a creare un intero genoma sintetico e a trasferirlo in una cellula batterica sostituendo il DNA naturale. È il primo batterio controllato da un genoma artificiale. E un microorganismo con geni sintetici, costruiti ad hoc, può essere utilizzato in modi molteplici. Può produrre nuovi carburanti. Può servire a creare nuovi vaccini. Può ripulire le acque o i suoli inquinati, digerendo sostanze tossiche e veleni. L'unico limite all'utilizzo di questa scoperta, ha spiegato Venter, è la nostra immaginazione.
So Craig Venter's announcement that he would sequence the whole genome by his privately formed company to say the least sort of disturbed us, because if it was largely sequenced by a private company, they could patent the DNA sequences. So a private group would sort of own the intellectual rights to the human genome. And I thought the human DNA is too important to be owned by anyone. It should be owned by humanity, available without cost. So we had a key meeting here in Cold Spring Harbor. Venter came, announced his plans. Francis Collins was here. And I took Collins aside and said, you have a choice between being a Chamberlain or a Churchill, you know. I compared Venter to Hitler, that, you know, Hitler wanted to own the world and Venter wanted to own the human genome. And I said, you got to act like Churchill. And Collins took on the challenge, but he was greatly aided that the Wellcome Trust sent their key person, Michael Morgan, and he made the announcement that the Wellcome Trust was going to double their money supporting it. So this then let Collins go to the US government and get more money. And so there was a race, which finally came to a draw. But we had made a very important decision, really. It was the Wellcome Trust that got it done several years before, which was the decision that the moment someone gets a complete DNA sequence, they deposit it on the web, so it's available to the whole world. So no one who got the sequence was going to get credit for it. in 2000, there was a famous occasion when at the White House, President Clinton welcomed both Venter and Collins, and I was there, but in the audience. And we've said that the Human Genome Project is done. Now, it wasn't finished, but it was going to afterwards be filling in gaps. So we declared a sort of that both sides had won. And the next 3 years was spent filling in the gaps. The English did 4 chromosomes, or maybe 6. The Department of Energy, which by then had developed a good program, they did 3 chromosomes. The French did a single chromosome. The Japanese did parts of their chromosome. I had been very mad at the Japanese earlier. I thought, they have all this money and they'll just use the human chromosome done by other people. They should pay for it. But the trouble with, if you understand the Japanese system, it takes about 10 years to make a decision. And so they finally made the decision to do it, but by that time, it was almost done. And so the Japanese did the part of 1 chromosome. There was by then a Chinese laboratory, Communist China, and they did a little bit. None of us knew how correct Obeko was in saying we needed it. It only became really useful when it was totally finished in 2003. And then you could begin to, the big job was look at the DNA and say, how many how many genes are there on our 24 different chromosomes? Once we had the human genome all out, we could begin to use it. And you could ask the question, say the question, in given cancer, does some part of the genome become more active than normally? Or, as we discovered, in cancer, some of the genes are present in 10 copies instead of 1. And in some cancers, you totally lose genes. Now we're sort of converging on a number of around 23,000 human genes. Before the project started, we thought there'd be about 100,000. And so 23,000 is just about 50% more than the DNA of a fly. So the number of human genes was smaller than anyone expected. It's still a large number when you have to take a look at 23,000 at the same time. And the other thing was found that given genes and through a process by which the RNA molecule gets cut and pasted, a given gene can be read in about 5 ways. So if you want to say, what's the number of distinct proteins that can exist in a human? The humans, about 100,000. That's the level of complexity. And an important technology was the bold move by people who were saying that we should use the techniques of the semiconductor industry to put DNA along DNA chips, the same sort of little fabricated piece which you use for semiconductors. You could put 100,000 pieces of DNA, and you can measure which of the genes were more or less accurate. We could finally do what Dobecco wanted us to do, which was to find the genes involved in human cancer and, say, compare breast cancer with lung cancer, etc. So we didn't really know what human cancer was until we had the human genome. And here at Cold Spring Harbor, we've focused on technologies by which we could, do it inexpensively and fast. Now that the Human Genome Project is done, you can't imagine doing science without it. I mean, we would be working 100 times slower if we hadn't done it. Maybe not 100, 1,000. So it led to what some people worry, big science. That you had bigger labs, and you had the technology in which it used to be, one scientist, one gene, study it. Now a given scientist is one scientist, 23,000 genes, and you got to look at them all. So that was really what we did for essentially, with the human genome, we could at last attack cancer.
Le malattie chiamate genetiche sono patologie causate da anomalie presenti nel genoma degli individui. Può trattarsi, per esempio, di un eccesso di cromosomi, come nel caso della sindrome di Down, oppure di altri tipi di mutazioni dei geni che danno origine a proteine modificate. Alcune portano nomi noti, come la talassemia, la fibrosi cistica, il daltonismo, l'emofilia. Quest'ultima, che induce un difetto nella coagulazione del sangue ed è causata da un gene presente sul cromosoma X, è anche conosciuta come malattia dei re, perché tra l'Ottocento e il Novecento si diffuse in diversi rami della nobiltà europea a partire dalla regina Vittoria, portatrice sana che la trasmise ai suoi eredi. Altre malattie che dipendono dai geni non si sviluppano automaticamente, ma solo in presenza di fattori scatenanti. Si parla in questo caso di predisposizione genetica. Ci sono poi patologie destinate a manifestarsi in età avanzata, come la corea di Huntington, che scrive nei geni, in base al numero di ripetizioni di una sequenza di lettere, l'età in cui colpirà il portatore. La conoscenza dettagliata del genoma umano è quindi decisiva per comprendere l'origine e i meccanismi di sviluppo delle patologie genetiche, nonché per ideare terapie di contrasto. Il progetto Genoma ha ottenuto le prime grandi vittorie proprio sul fronte delle malattie. Sono stati individuati i geni responsabili delle malattie ereditarie più comuni e di gran parte di quelle rare, così da consentire la diagnosi anche in fase prenatale. Inoltre avanza a grandi passi la terapia genica, un insieme di tecniche che permetteranno di sostituire i geni difettosi con versioni funzionanti, in modo da prevenire o curare i decorsi patologici. La rivoluzione genomica, con le sue incalcolabili applicazioni alla medicina, è appena cominciata.
When the human genome was completed in 2003, the cost had fallen tremendously lower than our estimates of $3 billion, and it was going down toward $30 million. which was still too great to sequence any single individual, except for Craig Venter. It turned out that his company had just sequenced himself, whereas the public program had taken 6 people from Buffalo, New York, and just mixed all their DNA together. So it wasn't the sequence of one person, it was a mixture. So Venter's company spent about $30 million doing his personal genome. At this time, there were efforts being made to make a big advance in DNA technology, and the first was by a company started near Yale University in New Haven, Connecticut, called 454. And 2 years after the Human Genome Project had been done, its president came to me and said they would like to sequence me with their new technology. And could they have my DNA? And I said yes, and they sent someone, collected my DNA, and I didn't hear anything for more than 2 years. And I thought, the technology doesn't work, but they called me up early in 2008 and said, we've sequenced half of you and are working with the Genome Center at Baylor University College of Medicine, and you'll be done in about 6 more weeks. And the cost of your genome would be about $1 million. So their technology had reduced, let's say, the cost by a factor of 50 or something like that. And in June of 2008, I went down to Houston, and they told me I had about 30 bad mutations. And before they looked at my DNA, I said to them, don't tell me what my DNA is like for the gene which is involved in increasing your risk to Alzheimer's disease. Because I didn't want to be told that 10 years from then I would probably be, non-functional. Otherwise, I said you can put my entire genome on the web. So they gave me a disk with all my sequence, and I came back here, and then we put it on the web so everyone could see it. I figured I could. I didn't really care what my genome said because I'd already lived a fairly full life. And but on the occasion, I said, start with only sequence old people. Because if you sequence someone age 20, he might get news he doesn't want to have, no. They could say, well, you're really at risk to a lot of cancer. Well, I wouldn't want to know it if you can't do anything about it. Since then, people have looked at my sequence and told me useful things that I didn't initially learn, such as I don't metabolize the drugs which control my blood pressure. So since I don't metabolize these drugs, I could quickly take too much of them. Once I knew my gene composition, instead of taking them once a day, I take them twice a week now. And so my genome, I think, has made me healthier. And I would recommend that more people do the same. The cost is falling very fast. 2 years ago, it was $1 million. It's down to $20,000 now. besides the company 454, no, there's at least 3 other serious companies. And at Cold Spring Harbor, we get machines from a company based on technology out of the Cambridge University chemistry department. So we're now sequencing victims of mental disease to see if we can't find the DNA changes which are causing them. We're even more excited by the possibility that the technology is moving very fast, and conceivably within 5 years, the cost of working out 3 billion letters will be down to $1,000. The technology for DNA is moving faster than the technology for computers in terms of the cost of obtaining the information. So the problem will soon move from sequencing someone to knowing how to interpret it. And so there is now being developed a sort of new breed of which is DNA bioinformatics, people who are clever with numbers, who can find ways to just look at the DNA molecule. So our limiting factor soon will not be machines, but people who are at heart mathematicians. The real limitation in moving forward is people who can analyze the data and not obtain it. And another thing we're doing now with personal genomes is we're beginning to do the complete DNA sequence of a cancer. Common sense will say, before you're treated for cancer, find out really what's wrong. Now we don't know enough, but say in 5 years, I wouldn't want to be treated with cancer before someone did my DNA sequence of the cancer and compared it with my normal sequence.
La divisione cellulare in un organismo sano è un processo rigorosamente controllato. I tumori si generano quando i meccanismi di controllo si inceppano e la proliferazione delle cellule avanza senza incontrare ostacoli. Nel genoma umano sono presenti una serie di interruttori che servono a stimolare e a fermare la divisione cellulare. Alcuni geni producono proteine che stimolano o aumentano la divisione delle cellule. Altri svolgono il compito opposto, servono a prevenirla. Quelli del primo gruppo, che svolgono importanti funzioni per la crescita e la rigenerazione dei tessuti, quando subiscono mutazioni, si chiamano oncogeni, cioè generatori di tumore. Quelli del secondo gruppo, invece, sono i soppressori tumorali. Quando comincia a dividersi, la cellula si trova di fronte a un posto di blocco che le impedisce di proseguire se non ha tutte le carte in regola. Ma se, a causa di una mutazione, gli oncogeni si scatenano e i soppressori sono posti fuori uso, il tumore può superare il blocco e avanzare indisturbato. La situazione è simile a quella di un'automobile con acceleratore e freno danneggiati. Il pedale dell'acceleratore bloccato nella posizione attiva e quello del freno rotto. Per proseguire nella sua crescita, il tumore deve però superare un posto di blocco ancora più rigido, presidiato da un gene chiamato TP53, che quando riconosce un comportamento anomalo nelle cellule, le spinge all'autosoppressione, in pratica al suicidio. È quando questo soppressore subisce mutazioni che il decorso del tumore diventa letale. Il gene TP53 è il custode del genoma, la più potente arma contro il cancro di cui dispone l'organismo. Le attuali terapie contro questa malattia si basano sulla sua stimolazione. Come aveva intuito già nel 1986 il premio Nobel Renato Dulbecco, conoscere il genoma umano è il solo modo per sconfiggere il cancro.
The day the announcement was made that I was going to lead the Human Genome Project, I had a press interview. And so I had to say something, and I said, we're going to spend 3% of our money pursuing the ethical and social implications of DNA knowledge. Because of the earlier sort of efforts of, particularly under Germany, of using genetics, or in that case, lack of genetic knowledge, to say some people are better than others. People were afraid if you got DNA information, you could discover that some people were less healthy than others, and you wouldn't give them jobs, or they couldn't get insurance. And so there was sort of apprehension, both of sort of personal apprehension that you'll find out things that you really don't want to know. I didn't think we'd have any real problems until we reached the level of the personal genome. But now we're there. So how are we going to deal with these issues? One way is that our Congress recently passed a law which said you cannot withhold health insurance from anyone by saying that their DNA puts them at risk. So that was very important, because now people will check themselves in some cases to see if they're at risk. A sufficient number of genetic diseases are common enough so that you have a finite chance of having a child with a genetic disease. And as a result of the Human Genome Project, it'll soon be possible to, again, a sort of DNA chip, which will say whether you're carrying one copy of a bad gene, which if you would get from both parents would make the child very sick. This is now already being done for a set of genetic diseases which preferentially occur in Ashkenazi Jewish populations. This is a social group where marriages are arranged. And so the religious authorities essentially do not let marriages occur without them both being, the couples being genetically tested, and then just saying you shouldn't have children, marry someone else. Now I'm very excited by the fact that these tests will now be given, I think, soon to any American or any country who wants them. And if you find you carry single copies of bad genes, have your partner tested to see whether you have it. Then you could have two choices. You just don't marry someone who has the same thing. Or there's a new technique called pre-implantation genetic diagnosis, by which you would take an 8-cell embryo test to see whether the embryo you have one chance in 4, whether the embryo will be healthy or diseased. So one of my long-term dreams is that we will soon be able to find some of the genes responsible for hereditary mental illness. The manic depressive disease just moves through families. And you'd like to be able to remove that curse. So if you knew the gene that was bad, by pre-implantation genetic diagnosis, you could be sure that the child was healthy. And I think it's very irrational that you shouldn't take positive steps to have healthy children, as if you're interfering with God. It's just common sense, if not socially responsible, of trying to ensure that your children will be healthy. Because if the child was sick, you're responsible. I would feel very guilty of not doing it. There are religious groups who say you shouldn't interfere with God's will. You know, how they know what God's will is another matter, to say the least. And so I just don't think about God. I would just think about what, will the child be healthy or not healthy? So my own impression is that the sort of ethical implications of the genome have sort of been overblown. It'll just be accepted that genetics will be like any other branch of medicine. Its aim will be to try and let people have healthy lives and not diseased lives. A major problem we'll face is that the 3 billion letters of any given human message means it's inherently complicated. So the average person wouldn't be able himself to look at his genome. His doctor wouldn't be able to. So there's going to have to come into existences not only companies that sequence your DNA, but then look at it and say what parts of it are good, and then inform your doctor what the DNA message is, and then the doctor can decide whether to tell you this information, because we want to keep people from unnecessarily worrying. You know, to say that you have a 5% greater chance of getting cancer, that doesn't help you. If you would say, unless you do this, you'll get cancer, you should know that. So there's got to be a lot of wisdom, combination of, you know, experience and knowledge. And so those people who will be wise and experienced will be better able to use the genetic knowledge. We should all know the basic facts of genetics, so that when we're told something, we have some freedom of choice as to how to react. The most common one now, which predates DNA, is Down syndrome, where you have 3 copies of chromosome 21. It leads to children with a lot of physical abnormality, a much foreshortened life, and a lower IQ. Most women want to be tested to see whether they're at risk, and if they find they're at risk, most of them decide not to have that child. So in the United States, I think the frequency of children born with Down's disease may be as low as 20% of what it was, which means that the average person actually behaves and they're dictated not by what their religion tells them, but just they don't want to raise a child who doesn't have an equal chance of having a successful life. And I think when we're told things about other genetic diseases, we'll behave in the same way. Who wants to have a child whose life will be blighted by a serious mental disease? You don't want it. So finally, we will have a healthier human population as a result of the genetic knowledge. And I take quiet satisfaction that I got the Human Genome Project started. The DNA structure, I wasn't that important. It was going to be found by someone. I was just lucky enough to have found it with Crick. The Human Genome Project might not have started for 10 more years, so to that extent, I've really changed society.
In passato, le coppie che presentavano un alto rischio di trasmettere ai figli una malattia ereditaria si trovavano per lo più di fronte alla scelta tra astenersi dall'avere figli e procreare sperando nella buona sorte. Oggi le tecniche mediche permettono di intervenire a vari livelli nel processo di sviluppo del feto, potendo di fatto prevenire la nascita di un bambino afflitto da gravi problemi di salute. L'intervento più comune è la diagnosi prenatale, che permette di identificare anomalie genetiche nelle prime settimane di gravidanza, prelevando alcune cellule del feto e analizzando il DNA. Per i genitori, nei casi in cui il nascituro si scopre malato, si pone l'alternativa tra proseguire la gravidanza o procedere all'interruzione terapeutica. Una scelta difficile, che solleva questioni etiche e religiose, alimentando un perenne dibattito nell'opinione pubblica. Le coppie ad elevato rischio riproduttivo possono però anche optare per un intervento alternativo, che li mette al riparo dall'eventualità di questa decisione. Grazie alle tecniche di fertilizzazione in vitro si possono infatti analizzare le cellule embrionali anche prima del loro impianto nell'utero. Si chiama diagnosi genetica preimpianto. I genitori iniziano un trattamento di procreazione assistita per fertilizzare gli ovociti materni con gli spermatozoi paterni. Si prelevano poi una o due cellule dall'embrione ai primi stadi di sviluppo e si analizza il DNA per rilevare il rischio di malattie specifiche. Se i geni non rilevano patologie genetiche, l'embrione viene trasferito nell'utero e la gravidanza comincia il suo corso normale. Ben diverso è il caso dei designer babies, l'intervento in vitro sui geni delle cellule embrionali per modificare il sesso del nascituro, un fenomeno in aumento che pone nuovi interrogativi etici sulla liceità degli interventi non terapeutici.
The ability to sequence DNA fast and cheaply will have the added benefit that we'll be able to really understand domestication, how you can start with a wild animal and produce so many different products or different forms of life from starting a simple one. And perhaps the most interesting one are dogs. uh, which became domesticated, we're not sure whether it was 10,000 years ago, from wolves. So what were the mutations? And by now we know that it's really only about 5 genes involved. And so differences in 5 genes explain most of the differences in all the dogs we have. Of course, besides shape, we're really very interested in how genes control behavior. And so whether your dog is gentle or violent, that has a genetic cause. To me, the most interesting thing will be using genetics to try and dissect human behavior. We could say, well, why, what makes a dog gentle? But the more important thing is what makes a dog violent and, want to bite you. And, will the genes involved in, or the major gene involved in violence in dogs be the same as involved in human violence? If I had to bet, I would say yes. And which then leads to a lot of complicated legal issues. If someone is born, which you could say born evil, and I could use the word, some people are just born bad. How should you treat them? If they really have no choice but to be violent, should you still penalize them? Should you put them in a mental institution? Should you lock them up? And with time, could you predict human behavior? So you look at someone's DNA and just say, he's going to be bad. And I don't know the answer. My gut reaction, which a lot of people is, if they're really bad, put them in prison and don't let them be free in human society. But, other people say you shouldn't penalize for something which they have no freedom. A lot of people would say, and people are capable of redemption. That there's no inherently evil person. But if we have an inherently evil dog, why not an inherently evil?
Ogni individuo ha delle caratteristiche caratteriali relativamente stabili che lo distinguono dagli altri e che chiamiamo personalità. I modi in cui si plasmano quei tratti che rendono ogni individuo un essere unico e irripetibile affascinano da sempre filosofi e scienziati. Sono i fattori genetici e quelli ambientali a influire maggiormente sull'organizzazione complessiva delle singole personalità. Oggi si sa che geni e ambiente sono entrambi decisivi e che solo la loro combinazione determina ciò che ognuno di noi è. Ma l'esplorazione del genoma umano ha permesso di comprendere come siano scritti nei nostri geni tratti della personalità decisivi come l'introversione o l'estroversione, la riluttanza ad affrontare novità o la passione per l'avventura. Uno dei geni più interessanti è il DRD4, Dopamine Receptor D4, che si trova sul cromosoma 11. Questo gene stabilisce, in base al numero di ripetizioni di una sequenza di basi che contiene, quanto il soggetto sarà ricettivo alla dopamina in determinate regioni cerebrali. La dopamina è un neurotrasmettitore fondamentale per la comunicazione tra i neuroni. Quantità insufficienti di dopamina provocano malattie come il morbo di Parkinson. Quantità eccessive, invece, possono causare la schizofrenia. Tra i due estremi si collocano tutti gli esseri umani sani, in cui una maggiore o minore ricettività alla dopamina non causa malattie, ma tratti peculiari della personalità. Un soggetto più ricettivo si appaga più rapidamente degli stimoli del mondo esterno e tende a diventare abitudinario, mentre uno meno ricettivo ha bisogno di uno stile di vita più avventuroso, di esperienze sempre nuove. Questo non significa che i caratteri innati siano immutabili. L'educazione e l'ambiente sociale possono modificarli. Restano però scritti nel nostro DNA alcuni tratti inconfondibili del nostro temperamento.
But if you look at it just from the practical viewpoint, uh, if you had a violent criminal and looked at his DNA and said, yeah, he looks evil or he looks violent in some way, then you would never release him from prison. Whereas the sort of traditional Christian message was, he will become good in prison and therefore he should be a chance for a second freedom. Okay, second chance. I think it just matters what genes you have. But then, you know, you'll say single genes can never have that much effect. With time, the criminal justice system will be strongly affected by genetic knowledge. That if the behavior of animals is controlled by genes, the behavior of humans will have some. And what you tend to find is genes win. That environment can, on the short term, but yeah, genes win out with time. We'll see. This last thing will be, you know, make some people just say, think Hitler-like. Forget Hitler. Ask yourself, what sort of knowledge do you need to run society better? And one is to be able to judge people better. And part of us is our genes. Part of us is, you know, what our parents were like, what our teachers were like. So those of us who know genes are important, don't say that the environment doesn't matter. As I get older, I just think we should use genetics, if we can use it, to make predictive knowledge. And I'm sure the average person will be, give the person a second chance.